AQA required practical is on osmosis. We will investigate the effect of different concentrations of salt solution on a plant tissue such as potato. What you will need for this practical are the following: potato, a cork borer, a ruler, 100 centimeter cube measuring cylinder, some boiling tubes on a test tube rack, and I've labelled them with the different concentrations of salt solution that I will be using. I'll also need a balance, scalpel, a forceps, white tile, some paper towel, different concentrations of salt solution. I have distilled water here, which will be my 0 molar salt solution. I have 0.2 molar salt solution, 0.4 molar salt solution, 0.6 molar salt solution, and 0.8 molar salt solution. First thing I'm going to do is use my cork borer to make some tubes of potato. So I have 5 tubes of potatoes, and I'm going to use my ruler to measure them the exact length, and then using a scalpel I'm going to cut them into the same length. So I'm going to make each of my tubes 3 centimeters in length. Making sure that the peel is not used, because osmosis will not take place if there's any peel. So here I have 5 tubes of potatoes, each the same length. What I'm going to do now is measure the mass of them on my balance. And I'm going to record the results on a table like this. So on this column I will put down the mass of the 5 pieces of potato. Once I've done that, I'm going to put all the potatoes into my boiling tube. And then measuring out 30 centimeter cube of my 0.0 molar salt solution. I'm going to put that into my first boiling tube. And continue to do that with the different concentrations of salt solution. So this is my 0.2 molar. I need the exact same amount, which is 30 centimeter cube. Continue to do that until I've done all of the concentrations. On each of my boiling tubes, I now have the potatoes and the different concentrations of salt solution. I'm going to leave that for 30 minutes and then I will come back to it. Now, using my tweezer, I'm going to take out the potato, pat dry it on some paper towel, Make sure it's in order so you don't get confused. Then I will measure the mass of these potatoes on my balance. And record the result on the table. So for the first concentration, which was 0 molar, I will have the first reading before I put it into the solution, the second reading after I take it out of the solution after 30 minutes, and then work out the change in mass. And that I will do for all the other concentrations of solution.